And the good thing is that it just sent me a mail telling me that he just get a neuroligin 4 mutation recently, and he was getting married very soon. So, 2 good news. So, but still we were there, I mean, with the neuroligin 4 story. Just 1 mutation, or in 1% of the population of autism, maybe less. But it was interesting because it was the first really functional mutation that we could find, and we could use it at iron freight to find other mutations. So that's what we have done. So there are the neuroligands there, and they can bind to other proteins. They can bind to Schenck, for example. It's a scaffolding protein at the synapse. And Christelle in my lab could find deletions, mutations. This little girl, she has a deletion of the Schenck-free gene, and she has typical autism. It was amazing that her brother here, he has 3 copies of Schenck-free. And he started to speak before the normal age, but he has a clear problem in social interaction and restrictive pattern of interest, and he has typical Asperger syndrome. So it looks like there is a gene dosage effect, with one girl missing 1 copy with very severe autism, and her brother with 3 copies with Asperger syndrome. So that was the second gene, and then we looked at others. I mean, neuroligands are there, and they can bind to the presynaptic side of the synapse to neurexins. And with the Autism Genome Project, we could find a deletion using this SNP array. And at that time, we were very happy with 10,000 SNPs. And we could find a deletion of the neurexin. And then this year, we used a SNP array that can interrogate 1 million of this SNP, and we could find new mutation deletions. I mean, a part of the genome which is out of the genome, and removing Schenck2, DLGAP2, SYNCAP1, PHD1. There are new synaptic proteins involved in autism. Some unpublished results. So we have also follow up to the Schenck2, and we could, it's a very complex genes with a lot of expression, splice isoforms. We could find a new CNV. So CNV is another genetic term. It's copy number variant, another deletion of the gene in one child, and some mutations. So, to summarize, we could find the neuroligands, very limited subset of individuals, but they could tell us a little bit more about what autism could be at the genetic level. We could find mutation in Schenck, in Schenck-free, in Schenck2, and neurexin-1 was the other one. So, to make it very simple, if you want to go from Institut Pasteur in Paris to Catania, you can be stopped here, you can be stopped here, or you can be stopped here. So it's probably the same pathway, but you are stopped at different levels. Okay? Okay. So what are the other genes, les autres gènes? So we are using this 1M SNP array, and we have new 450 patients that we are looking at carefully, and very soon, next year, we will get, if I have the funding, the patients, the exome, I mean, most of the sequence of the human genome. So just to show you how it looks like in a lab, you're all invited at the Pasteur Institute to come. And this is a software that we developed at the Institut Pasteur. So you can virtually, this is chromosome 11 of one child with autism. And then you can go here, and each dot here, it's a variation in your genome. So it's like a PlayStation. I spend my life now looking at this genome. And then you can see here that something happened. You don't need to be a geneticist to see that something happened. And here you have a child who lost 1, 2, 3, 4, 5 millions of base pair, I mean letters of the DNA, removing all these genes, and 1 or 2 of these genes are probably doing something in the development of autism in this child. So, using this new technique, we could find the old fellows. I mean, Neurligin 4 again, Sheng3 again, Sheng2, I told you, P9 is a new gene, C18 is a new gene, another very, very interesting gene, and chromosome 11, deleted sometime, duplicated in some cases. So, but finding a gene is really interesting, but it's really the beginning of the story, not the end. That's for sure for me, at least. So now you have to ask me the bad questions. Do you have functional evidence that your genes are doing something in the development of autism? And this is one of my answers. This is Neurligin 3. And if you overexpress a Neurligin 3 in a neuron, you will get all this little labeling here. And these tiny labelings are synaptic contact. And then you can do the same, but instead of overexpressing the Neurligin 3 wild type, you overexpress the one with the mutation that we found in the Swedish family. And you don't have to be a neurobiologist to see that there is no more synaptic contact, or very few synaptic contact, when you overexpress a Neurligin 3 with mutation. And we have done the same with Sheng3, and this is unpublished. We have the same with Sheng2, and I will have no time to go into details. This is in vitro in the culture dish. So what's going on in vivo? So what we have done is to find the genes in the mouse genome and then to mutate this gene in the mouse genome. And that's what we've done in collaboration with Niels Broos in Göttingen, in Max Planck. We knock out the gene, and then we could look at the mice. The mice were almost normal. There was some slight reduction of the cerebellum, some reduced social interaction. I mean, it's published. You can go to the publication if you want to have details. Very slight reduced social interaction, but still, it's really interesting. And also something which is, maybe you don't know, but mice can sing. Mice can sing. But they sing in the ultrasound frequency, so that's why you never heard a mice singing, probably. And so this is the mice, the wild-type mice singing, creak, 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 like this. So this is the wild-type here, and this is the Neurligin 4, with really the vocalization are really fewer than the wild type. So we are looking at now, we are recording these mice to see what's going on when this deficit appear. And also unpublished results, but we are very, very happy to have new models. And so let me introduce you to the Sheng2 knockout mice. This is the first time I show this. And so it's really unpublished, but we have already very nice results, showing that the mice have higher anxiety when you challenge them with the white or black box. So the Sheng2 wild type, they stay in the white part, and the Sheng2 minus, they goes directly to the dark phase of the box. So it's really significant. And this Sheng2 here, you can't see. We have to wait 20 minutes or 27 minutes to see that it's really hyperactive mice. So it's a really interesting phenotype. So there are these synaptic parts. So we have fragmentation and CNVs, so copy number variants in Neurligin, Schenk, Neurexin, all these genes. But probably they participate to the same pathway, probably altering synaptogenesis, imbalance between synaptic currents, GABA glutamate. And we have animal models now to try to understand, at least in the mouse, what's going on when these genes are mutated, and can we reverse the phenotype. So one thing that was interesting also was the clocks. So let me introduce the sleep-wake cycle of a newborn baby. So to my knowledge, I have 2 kids, and I remember very well, in the beginning there was kids were black when they are sleeping and white when they are not sleeping. And if you remember, it's a chaos. There's babies sleeping, not sleeping, sleeping, not sleeping, sleeping. And then it's maybe worse because the child is sleeping, not sleeping, sleeping, and it's free running. because your biological clock is a little bit more than 24 hours. So if you're in a cave, you're going to bed a little bit later each day of the week. And then one day, it's magic, the child is sleeping during the night, and the parents also are sleeping during the night. And the child is awake during the day, sleeping during the night. And so what is this time giver? And one of the time giver is melatonin. And melatonin is high during the night and low during the day, high during the night and low during the day. And it is synthesized mainly in the pineal gland, in the brain. And Descartes, the French philosopher, thought that the pineal gland was a seat of soul. So I think my results don't go to this phase, but it's interesting.